Mankind has not yet succeeded in inventing medicine for all diseases. Although the development of medicine has always been closely associated with the development of the pharmaceutical industry and surgical techniques, for a long time chemical drugs have often given rise to additional unnecessary problems, after eliminating previous ones. Eventually, it became clear that treatment ideology must undergo changes, but it seems that scientists have found a solution that could revolutionize medicine. A human being is composed of cells which are regulated by signaling molecules. These are biologically active compounds of different classes that send commands to cells, telling them what they need to do. So in today's high-tech world, a person is regenerated naturally instead of artificially. Therefore, a cure using internal resources of the human body is today one of the strategic directions to which scientists in different fields have been engaged. The cells that make up human organs are in fact complex systems. Their subunits, known as organelles, make them function. Advanced technology has reached such a level that an organ or even a body can be grown out of individual cells. The most promising are the stem cells, because they are primary, and they can be transmogrified into specific cells under certain conditions. So, scientists have concluded that with the help of such cells, damaged parts of the human body can be replenished and regenerated. This is an important distinctive feature, and if we could replicate it, then we would be able to treat that which in principle cannot be treated, and then move forward from treating to curing. However, stem cells can also be different. The Institute of Molecular Biology and Genetics decided to focus on so-called mesenchymal stem cells. Bone, cartilaginous and fat cells are formed out of them. Also, certain signaling molecules that trigger the mechanism of recovery of tissue cells are synthesized in them. This property lies at the root of development that may become a new generation therapeutic instrument. We work directly with the institutes of the Academy of Medical Sciences of Ukraine. Apart from advanced research centers, it provides room for replication and replenishment, and vivaria to work with animals. Scientists are seeking ways to restore the central nervous system in the human body together with the Institute of Neurosurgery. That is one of the most complex body systems, which is the most difficult to treat and damage to which is in most cases irreversible. Multiple sclerosis, for example, it is still believed an autoimmune disease when the immune system destroys its own tissues, namely myelin, a special layer protecting the nerve fibers of the brain and spinal cord. If it is damaged, the connection between the brain and other parts of the body is disrupted. The disease causes loss of vision and control over extremities. A person frequently faints, which is additionally accompanied by mental and psychological problems. There is currently no effective medication to treat multiple sclerosis. All hope lies with the research of molecular biologists. They were able to show that the infusion of stem cells can restore the functions of the central nervous system. A sick animal was losing its mobility. The conductivity of the central nervous system has slowly been in a state of degradation. If that happened to a human, they would be completely disabled. When we injected the stem cells, even after the damage had already developed, we witnessed full recovery. Now we need to test this technique on humans. Just like the technology of recovery of the mucous coating of the nasal cavity, this is part of a joint project with the Institute of Otolaryngology. The peculiarity of the nasal mucosa is that it has many layers. The top one is the so-called ciliated epithelium. If the mucosa is destroyed due to inflammation, it practically does not return to its previous state. But with the help of stem cells, the mucosa can be restored. This has been proven through experiments on mice. After injecting mesenchymal stem cells, full recovery is observed and is confirmed not only by the overall condition of the animal and its behavior, but also the histological sections. The mucosa and ciliated epithelium are restored. In other words, complete recovery is observed. The liver is a very vulnerable organ. It can be severely damaged by leading an unhealthy lifestyle, taking medications intended for other diseases, inhaling industrial emissions, catching an infection, or eating unhealthy food. All this affects this organ. In the beginning, fibrosis starts to develop. Fibrosis is the formation of excess fibrous connective tissue in the liver. Just as scratches or wounds heal by the means of connective tissues, so a scar forms. The liver tries to heal itself in the same way. This can lead to such deadly diseases as cirrhosis or liver cancer. So far, transplantation has been the most common applied method of treatment. Obviously, this method is not available or affordable for everyone. Not only because it is quite expensive, but there are just not enough donors. Around 2,500 patients in Ukraine need liver transplantation annually. The figure is even more significant on a global scale. So, when scientists found out about the potential of stem cells, restoration of the liver tissue has become one of the main fields of research for scientists around the world. The aim of our study was to develop ways of liver repair by MSC transplantation, in this case the umbilical cord. Mesenchymal stem cells are contained in the tissue which forms an umbilical cord. Interestingly, MSCs can be used not only for the person from whom they were removed, but for anyone else, even for animals. Cells with this property are called allogeneic. They facilitate research as they allow for testing therapeutic techniques on animals first. For example, liver damage in rats is very similar to the damage to the liver in human beings. It starts with fibrosis, which later develops into cirrhosis of the liver. The liver contains connective tissues, so-called nodules. The whole liver tissue is covered with these nodules. The liver gets completely disorganized and its function changes accordingly. This disease, in most cases, ultimately leads to death. But researchers save animals by injecting mesenchymal stem cells taken from the human umbilical cord. The section of a liver under a microscope shows that its tissue can almost completely be restored. We have been working hard to find out what dosages to use and how to inject these cells. And we have received excellent results. As seen in the image, the connective tissue dissolves and the cirrhosis-affected liver initially transitions to fibrosis and later, gradually and almost fully, recovers. Scientists have worked out the technology and established a protocol. Now it will be further tested in hospitals. If the trials prove to be successful, then the deadly cirrhosis could be treated by simply injecting stem cells into the vein. To develop treatment, we must understand the nature of a disease and be able to recognize it on time. Therefore, employees of the Department of Regulatory Mechanisms in Cells are gaining insight into microbiology. They are studying the behavior of bacteria in different conditions and different ecological niches. As part of this work, we study biofilms, in particular the method of aggravation of microbial cells, how they behave in a wound, and the mechanisms involved in the formation of biofilms. In other words, how bacteria behave, how they communicate, how they cooperate, and how they form biofilms. This is not just scientific curiosity. The accumulated information gives many possibilities, in particular in diagnosing infections. Scientists have focused primarily on two. The first are staphylococci. There are many varieties of them, and they are very insidious. They can quietly reside on the skin or mucous membranes. But as soon as certain conditions arise, they begin developing rapidly and excrete toxic substances, causing purulent inflammation. A recent surgery or chronic disease exacerbation provide the most favorable conditions when the body and immune system are weakened. Pregnant women and infants are also at high risk of contracting this disease. The worst is that staphylococci are resistant to most antibiotics, and choosing an effective drug to combat them, you must know exactly what kind of staphylococcus has caused this infection. Our task was to develop a test system that would allow diagnosing the presence of Staphylococcus infection relatively quickly, within 3 hours, and at the same time to answer the question of which antibiotic to use to stop the inflammation. This test has shown brilliant results, and some Ukrainian laboratories are already using this method. And the institute has launched a new project: the development of diagnosis for another serious illness, Pneumocystis pneumonia. Pneumocystis is not a bacterium, but a body close to fungi. Therefore, it is difficult to cultivate. It is diagnosed only in specialized laboratories. It appears in people with very low immunity, patients who have received donor organs and patients suffering from AIDS in particular, because they have to take so-called immunosuppressive drugs to prevent the immune system from fighting the alien body. Here we want to combine classical and modern molecular microbiology approaches. Therefore, our tests are based not on culturing microorganisms, but on examining genetic material in infections. Blood, skin or mucosa samples are suitable for analysis. The test was specially developed to detect a DNA pathogen, even if its quantity is insignificant, simply because it's based on the so-called PCR, polymerase chain reaction. It is a technique used to amplify a single copy or a few copies of a fragment of a DNA. We have developed these reagents, we have combined them properly, and we have developed a manual and the order of reagent amplification. Any laboratory interested in the diagnosis of infections can apply this test for diagnosing Staphylococcus and, hopefully, pneumocystis in the foreseeable future. The Institute of Molecular Biology and Genetics undertakes the most challenging tasks, for the most of which advanced solutions have yet to be found. And the years of experience of fundamental developments allow the Institute to keep pace with the world trends. When mankind eventually rises to a new level of fighting diseases, Ukrainian scientists will undoubtedly stick to this path.